Good afternoon, everybody. I would like to thank the organizers for giving me this great opportunity to come here and present our work. And I understand that it's the last day of the conference and I'm the last one before lunch. So I really appreciate all of you sticking around, and hopefully my talk will give you a good appetite, not only for lunch, but also for some more scientific discussions afterwards. So today I want to talk about a new framework that we've been working on to construct atomic resolution structurally resolved interactome networks, which hopefully by the end of my talk I can convince you that will greatly improve our understanding of molecular mechanisms for human disease mutations. And speak of disease mutations, we actually have this great asymmetry. On the one hand, we have an explosion of sequencing data with tens of millions of variants that has been identified so far. You can see this red line is the number of SNPs in dbSNP is going almost straight up. But unfortunately, we don't have a functional understanding for the vast majority of these identified SNPs. As a result, the number of disease-associated genes and their mutations, the blue and green line, it's not growing nearly as fast. They're more than 10 times slower than the number of SNPs that are growing. And even worse, the number of FDA-approved drugs, the yellow line here, is going really along the x-axis, is not really growing at all. And for many of these disease-associated genes and mutations, we don't know how they cause the disease or if they are causal in the first place, not merely correlations. So what this means is, although, of course, sequencing data is very important and we should get more of the sequencing data, but sequencing data alone is not enough, and we need to study really their function and their molecular mechanisms to help us make use of these sequencing data. So in the past decade or so, people have started to use the interactome network to understand, sort of function and the molecular impact of disease mutations. And it has been really helpful, but I think you will all agree with me that the success is still very limited. And one main reason for this limitation, I think it's because there's this dichotomy in network biology between network modeling and biological understanding, right? So far, when people talk about networks, they will show a hairball-like picture like this with thousands of proteins represented as graph theoretical nodes here. They're all the same, absolutely no difference. These are mathematical dots, right? They have no shape, no structure, no volume. But on the other hand, in biology, we also know that different proteins have distinct structures, and the unique structure of each protein fundamentally determines its unique function. In fact, structural biology is an integral part of biology, and many Nobel Prize actually came from for structural biologists. And it takes a lot of hard work, but I think so far a network biology field has been somewhat underutilizing the structural information. Don't get me wrong, these kind of graph theoretical representation of networks are, has been and will always be very, very important. And my lab still uses these abstract network models to do all kinds of analyses. But for certain analyses, like the ones that I will be talking about today, I believe that adding the structural information is essential for their success. Therefore, to bridge network with biology, we set up to integrate the supreme resolution of structural biology with the expansive scale of protein networks to construct structurally resolved protein networks, especially in relationship to human disease. So to do this, first of all, we try to get as many interactions as possible. We combine our own East to hybrid screens with all reported interactions in the literature by going through all major databases. A key here is that we use a series of filtering steps to ensure, number one, our interactions are high quality, and number two, the interactions we use are direct binding of each other. Turns out, neither of these are actually necessarily true for many of the interactions reported in the literature. Number one, because of the nature of the methods, vast majority of the interactions reported in the literature are produced by especially pull-down mass spec assays. And most of these interactions are what we call co-complex associations. They are not direct binding of each other. And because we're interested in finding the binding interface between two interacting proteins, so these interactions, these co-complex associations are not applicable to our analysis, so they're out. And secondly, and this can be surprising to many people, but we have actually published a series of papers on this topic, is that we realized computationally and verified experimentally that many of the interactions reported in the literature are actually of lower quality. They're not reliable. So we use a series of filters to remove those, so only have direct binding and high quality interactions. So after that, we combine all of these interactions with all of the known co-crystal structures in the PDB. So for those that has co-crystal structures, finding the binding interface is quite straightforward. It's not easy, but it's quite straightforward. Unfortunately, less than 10% of all of these known interactions have a corresponding co-crystal structure. So for the, the other more than 90% of the interactions, we developed a homology modeling approach to build, computationally predict 3D models for these interactions. And at the end, so we have a 3D interactome network where not only we know, all of the proteins that interact directly with each other, but for each and every one of those interactions, we know their binding interface, where they bind. And it just provides a lot more information with a much higher resolution. And then we can map all of the known disease mutations onto this 3D interactome network. And it's not easy to build these 3D network interactomes. It takes a lot of effort. So we built two databases trying to disseminate our data. So hopefully to help you with your own research, we have HINT, where we have high quality interactome networks for a number of species, including human, and we report separately binary direct interactions versus co-complex associations. You can download either or both for your purposes. And then we have Instruct, where we try to use our homology modeling approach to structurally resolve the binding interface of these high-quality interactions as much as possible. And we constantly update our data. Within less than 2 years of the publication of our first paper, we have more than doubled our interactome size, our structurally resolved interactions, and all of these numbers. So I strongly urge you to check out our websites. But before I talk to you about how our 3D network can help us understand disease mutations, I just want to very briefly reiterate some very simple concepts. Mutations can be broadly divided into two categories. We have the in-frame mutation, missense mutations, short in-frame insertions and deletions, that most of them we believe will lead to local defects on the protein and therefore cause loss or gain of specific interactions. And then we also have this so-called truncating mutations, including nonsense stop codon mutation, out-of-frame indels, and splicing defects, which people believe that most of this will not lead to any protein products through nonsense mRNA-mediated decay, through misfolding, etc. And therefore, you will lose the whole protein, and you will cause basically loss of all of the interactions. In fact, in the literature, in many high-profile publications, these interactions are often called LOF, alleles corresponding to complete loss of function, right? You lose the whole protein product. So for now, let's focus on the in-frame mutations because these seems to be more specific and more interesting. But we'll come back to these LOF alleles later in my talk. So the first thing we did is we look at, within our 3D interactome network, we look at the distribution of the in-frame disease mutations and where they are with relationship with the interaction interface. Please note, in this paper, the first step, our resolution is at the domain level. So these are interface domains, right? So then we found that there is a very strong enrichment for in-frame disease mutations to be specifically in certain specific interaction interface domains, right? So that means it seems that these disease mutations are specifically disrupting specific interactions, and understanding these specific disruptions is very important in understanding the pathogenesis of many of these disease mutations and genes. So, but some people say, well, but domains are functional and structural units of proteins, so maybe this enrichment is not because they are interface domains, but because they're just domains. So we did the control where we look at any other domain that are not used at the interface domain, and there is absolutely no enrichment whatsoever. Again, showing this enrichment in interface is very, very specific. And thanks to the anonymous reviewer, we also did this control calculation where we take all of the non-synonymous SNPs in dbSNP, vast majority of which are believed to be benign, and we did the same calculation. There is no enrichment wherever, again showing this enrichment is very specific. So, and then actually we look at the subset of the interactome that has corresponding co-crystals where, again, very straightforward, we can identify the specific interface residue because these sort of enrichment of disease mutations on the interface domain at the time, we believe, and in the literature many people believe, really the signal comes from the interface residue, the specific interface residues that are really making contact between the two proteins. But at the time, we just don't have the resolution to look at them, right? So now we focus only on the co-crystal structures, and we can identify specific interface residues, and we repeated the calculation. And I expect to see the signal only exist for the interface residues. But to my surprise, what we found is that there is enrichment both for the real interface residues, but as well as other residues that are in the same interacting domain, but they are not actually the interface, right? So meaning that both of these residues in the interacting, the whole domain is really important. And that sort of makes sense if you think about, again, we know that domains are structural and functional units of the protein. And if you disrupt other residues in the domain, you can definitely cause change the conformation of this interacting domain and disrupt interaction. So that's, again, that's actually quite surprising compared to the common hotspot, all of that corresponding to only interface residues. So that's interesting. And actually, so just one more thing, we actually did other analysis in this paper looking at the relationship of how damaging the disease mutations are versus their relationship with the strength of the interaction. And we also look at other structural and physiochemical features of the disease mutations and compare them with changes occurred through the evolution. Unfortunately, I don't have time to go through any of this, but I urge you to read our paper. Is on my lab website. So we look at we build 3D sort of models for interactions, and it's seen to be correlating to the specificity of disease mutations. Doesn't really work, right? So we want to experimentally verify our computational predictions. So as a proof of principle, so we pick this interaction between MH1 and PMS2, two well-known protein that involves in DNA mismatch repair. And there's a ton of mutations on these two proteins known to relate to various diseases, especially colorectal cancer. But despite that it's a well-known interaction, there is no co-crystal structure. So we built this, use our homology modeling approach from MH1 homodimer, and based on our prediction, we predicted that these two domains on MH1 and PMS2 are most likely the interface domain for this interaction. And as I said, there are literally hundreds of mutations on MH1 that are in the literature reported as causing colorectal cancer, right? And there's really no difference between them. But when we look at this, there is a very strong enrichment for these mutations to be on these two domains. But because there are literally hundreds of them, so there are not small number of them outside of these two domains. So to verify whether our predictions are correct, we randomly picked 3 red mutations that are on these two domains and 3 blue mutations that are not on these two domains that we predicted to be the interface domain. And again, these 6 mutations in the literature, there is absolutely no difference between them, right? And our prediction will be these 3 red mutations will disrupt this interaction, and these 3 blue mutations will unlikely to disrupt the interaction if our 3D network model is correct. And we tested with Y2H. As you can see, only the 3 red mutations disrupt the interaction. The 3 blue ones does not impact on the interaction at all. Of course, this is only one experiment. I'm not going to claim these 3 blue ones are passenger mutations, whatnot. But if I were to put money into follow up on any of these mutations, I would choose one of these red ones, not one of the blue ones, right? So that means that our 3D network approach can, is valid and can help us determine driver or causal mutations and genes, right? Not only for cancer, but for any disease. One major roadblock in human genetics is really the complex genotype-to-phenotype relationships, right? And one of such is so-called locus heterogeneity. That is, that mutations in many different genes can cause seemingly clinically same disorders, right? And so one main goal is trying to sort of predict what mutations in what genes can cause this particular disease using known disease mutation information. And this is where actually this guilt by association principle come into play. This has been more than 14 years people have been using this, and it's again has been really helpful. And so what this says is that if mutations on 2 interacting proteins will be more likely to cause the same disease, right? So we decide to sort of verify this, which has been published in the literature over and over again. And so what we did is we look at pairs of mutations, right? Pairs of mutations on 2 proteins, on 2 different proteins, and either the 2 proteins are interacting or they're just random 2 proteins we picked. And we look at the fraction of these mutation pairs causing the same disease, right? We're trying to validate, and this is not our results. We're trying to recapitulate what's already published. And turns out, not surprisingly, that the literature is right, that there is a very strong enrichment for mutations on interacting proteins to cause the same disease. So that's good. But if you look at it, it's only about 10%, meaning that more than 90% of the mutation pairs, even if they're on the interacting protein, they won't cause the same disease, right? So it's not a great predictive feature, right? And my background is in machine learning. So we say, okay, whether our 3D network model can improve this, right? So now we split the first bar into the 2 mutations on 2 interacting proteins, and if they located in the corresponding interface that are bridging that interaction versus they're on the interacting protein, but they're somewhere else on the protein, right? And we're very happy to find that actually our 3D network model can significantly improve sort of the performance of guilt by association, right? If they're on the corresponding interface, it's much more likely to cause the same disease. So we're very happy. Our paper is published, and it's actually quite well received. But some people told me, well, you made a big fuss about how just using guilt by association only successful 10% of the time, and your 3D network really greatly improves it. But if you look at the specific percentage, it's less than 15%, right? Meaning that over 85% of the cases still doesn't work, right? That's not great predictive performance either, which is absolutely true, right? So I thought, okay, can we further improve this? Well, if in genetics, mutations are not only divided by inferring versus truncating, but mutations actually have different inheritance mode. You have recessive mutations that you need 2 copies to see the disease phenotype, and you have the dominant mutations where you only need 1 copy. So we say, okay, let's divide the mutations by their inheritance mode. First, we'll look at recessive inferring mutations. And boom, this result is really surprising, pleasant surprise to me. For the recessive in-frame mutations, close to 90% of them, if they're on the corresponding binding interface of two interacting proteins, they will cause the same disease, versus if they're somewhere else on the same interacting proteins. And that is great predictive power, right? And we're not even doing any learning here. This is raw data, right? So we're very happy about this. And surprisingly, when we look at dominant in-frame mutations, we found, very surprisingly, that guilt-by association actually doesn't really apply at all, right? You don't see any really much power here. So, we thought, why is this? Well, if you think about it, many of the recessive mutations are because of loss of function. So if you lose an existing function, the existing interaction and the existing interface, of course, play a role of what, losing that function means. But a lot of the dominant mutations are gain of function mutations. So if you gain a brand new function, what existed before really doesn't help you understand, gaining the new function, what it means, right? So to check this, so that's our hypothesis. Maybe it has something to do with loss or gain of function, the nature of the mutation. So to check that, for the dominant mutations, actually there is people have a very nice prediction, with experimental data, but also prediction of haploinsufficiency leading to dominant mutations, or non-haploinsufficiency, right? So for those haploinsufficiency causing dominant mutations, they are loss of function clearly, right? Because so we divided the dominant mutations into, haploinsufficient dominant mutations versus potentially non-haploinsufficient dominant mutations. And we found that for the haploinsufficient ones, guilt by association, now you can see the separation between on the interface versus somewhere else on the protein. Guilt by association start to work. So really, guilt by association does not apply to gain of function. I think this sort of indicates that our hypothesis is right. It's the gain of function mutations. And we have many, many algorithms out there trying to predict new disease genes, disease mutations using interaction networks. And I haven't seen they really integrate the inheritance mode or this loss or gain of function nature of the mutation. But we have, I hope clearly shown here, this is very important in your predictive performance. So we hope, in the future, people will incorporate that into their algorithms. And then, we decided to, let's take a look at, so these are all in-frame mutations. We said, okay, let's look at LOF mutations, right? To me, this is really just a control calculation. I did not expect anything to happen here. And what is our expected results? Well, because these mutations, people believe, and we believe at the time, will lead to no protein product wherever they are on the protein, right? Because they don't have any positional effects. So we expect the results should look really like dominant in-frame mutations, because you will not have any positional effects, whether it's on the interface. For this truncating, we use on or before, right? Because on or before it disrupts that interface. On or before that interface or somewhere else on the protein, right? Really, you should have no positional effects if vast majority of LOF mutations are truly complete loss of function leading to no protein product, right? But the result, again, is a surprise, right? We found even for these so-called LOF mutations, over 60% of them, if they're on or before the corresponding binding interface on the interacting protein, they will cause the same disease, versus, less than about 15% if they're elsewhere on the protein, right? Showing a very strong positional effects. And that is very surprising, because what this means is that there is a significant portion of truncating alleles that can actually give rise to functional products, right? And so sort of that's contradictory to the common belief. And if we look at the dominant ones, that's not surprising. Dominant, doesn't apply to dominant guilt by association. But if you were me, I think you would still be very puzzled by this, right? By this strong positional effects for LOF alleles. So I thought about this. I thought, okay, maybe the allele, the LOF alleles that gave you this signal, this strong positional signal, are all at the end of the C-terminal of the protein, right? So if they're at the end, they only shave off a few amino acids. I can imagine that protein survive and have a peptide, right? Have a functional peptide. So what we decided to do is only focus on these first two bars, because the key is whether we see the positional effects, right? And we don't expect to see positional effects. And we divided all of the proteins carrying LOF alleles into 3 equal parts: the first third, middle third, and last third, right? Again, my expected result is this strong positional effects, the difference between these two bars, are only going to occur at the last third if it's going to happen, it's only going to happen at the last third of the protein, not at the beginning of the protein, right? I was again wrong. And we found that even, in the first third of, even if the LOF is in the first third of the protein, meaning that it will cause the protein to lose more than 2/3 of its length, a significant portion of that can still give rise to a functional product, right? Again, we have, the sequencing is going on right now, and we have many algorithms analyzing sequencing data, trying to relate them to various disease. And as far as I've seen, most people will just, when they see a LOF, they will just say, oh, it's completely lost function. Again, our results show if you do that, you may, come to sort of misleading results. Just very quickly, we just, again, proof of principle. We have this TRIM27 interacts with these 3 proteins, and we use again, there's no crystal structure, use our 3D network modeling. We found that the 2 proteins interact at the first interface domain, the third SERPA protein interact at the sort of C-terminal interaction domain. Using wild type, all 3 interacts. There is actually a small out-of-frame deletion right before the second interface that actually causes ovarian cancer, associated with ovarian cancer. We cloned in that mutation. We found it specifically caused disruption of the third interaction, but the first 2 are completely intact, right? Again, so shows. And then this actually also applies to all of the ESP 6500 SNPs. You can see the rarer the allele, the more enriched they are on these binding interface, right? Just to very quickly summarize, we've constructed a 3D atomic resolution protein-protein interaction network for human disease, and we actually not only know their interaction, but we know exactly their binding interface. We found that in-frame disease mutations are very specifically enriched at that interface, and the interface residues, as well as other residues in the same domain, are equally important in terms of understanding the potential disruptions of the disease mutations and their distribution. And we also found that we can really dissect complex genotype-to-phenotype relationships through 3D interactome networks. Many other actual relationships reported in our paper that I don't have time to talk about. But really, 2 key things that I talked about today is that guilt by association works great for recessive mutations, but really not very good for dominant, especially gain-of-function mutations, and a significant portion of the truncating alleles can give rise to functional products. With that, I want to thank my lab, thank my collaborators, and thank my funding agency, and thank you for listening.